Yeah, sure. I'm really delighted to introduce Emily Begnel, who will be giving us a talk today on gut virome ecology on a birth cohort in Kenya. So Emily is currently a PhD student in the Department of Epidemiology. She's been a study coordinator on the Linda Kizazi study, which you'll hear about today, since 2018. I've had the pleasure of working with Emily for many, many years. Before that, actually, as an MPH student in epidemiology, and she was one of my first TAs for SPH 480 in the undergraduate program. So it's been amazing to watch her professional and academic journey. And this work that she will show you today will demonstrate clearly her knowledge and expertise in both epidemiology and data analysis. So really excited. She's going to present one of her thesis aims today, and would love to get feedback on that as she's polishing this up for the thesis in manuscripts. So take it away, Emily.
Yeah, thanks so much. I'm really excited to present this data. It's still in a somewhat early stage, as we'll talk about. So as Gwen said, if there are any questions that don't seem to be addressed through this, or any big comments for feedback, please, I welcome them. So just as a quick overview, I'll do a little bit of background just to explain what the virome is and why we're interested in studying it in infants. And then I'll talk about the Linda Kizazi study, how I'm approaching this analysis. I'll show you some of my initial results that are looking at HIV exposure and the infant gut virome, and then some sort of next steps and key takeaways. So the virome is simply the collection of all viruses in the body. So our body has all sorts of microbiota, viruses, bacteria, fungus, protists, these archaea, all these things. And so the viruses is one component of what is collectively referred to as the microbiome. So when we're studying the virome, we're just studying specifically this viral part. Viruses are super diverse. There are prokaryotic viruses that infect bacteria, which I'll be referring to as bacteriophages or phages throughout the presentation. And then also eukaryotic viruses that infect us, and also these other eukaryotic organisms in our body. Viruses have a lot of shapes, structures, might have DNA or RNA genomes. They may be pathogenic, like the familiar ones that we know, like HIV, cytomegalovirus, flu. Or they might be commensal, they just sort of exist with us. And some might even be beneficial, as we're starting to kind of see in the field. Like the bacterial microbiome, the virome is different within different biological compartments. Here today, I'm going to focus specifically on the gut, which is so far the best studied compartment of the virome, but this is also being examined in blood and breast milk and genital fluids, all sorts of other, saliva is another one that's common. So anywhere you can study the sample, you can study the virome. And as we'll touch on, there are a lot of interactions between viruses and these other kingdoms, and the way that we understand those is we're still learning a lot about those and what that really means for our health and disease. But we know that will be sort of important to take away as we learn more and more about the virome. Also, just as a quick overview of how we study the virome. So virome comes, as I alluded to, out of some sort of biological specimen. For the gut virome, it's usually stool, although sometimes you see studies with rectal swabs. And you process the samples to extract the usually DNA, occasionally RNA, but most virome studies are looking at DNA viruses, which is one of the current limitations of the field. They're more stable in samples, and that's part of the big reason that we only look at DNA usually. But you take all of the DNA out of the samples, sequence them on lots of expensive, fancy lab equipment. And as you'll sort of gather from this presentation, my involvement in this pipeline of how we get through a virome analysis really is at this beginning stage where we're doing a study design, thinking about sampling, thinking about selecting samples for an analysis, and then again at the end when we're doing some of this bioinformatics work, and then also the statistical analyses. But the general idea is you take the DNA out, you sequence it on these platforms using these assays, you get out chunks of DNA that are called reads, and those are assembled through these bioinformatics methods, which are very statistics heavy. It's a whole discipline in of its own. There's a lot of really interesting things to read about it, so I won't go into detail there, but happy to answer any questions I might be able to about that are relevant to this presentation. But we process the data, try to basically group these sequences together into what are usually called contigs or contiguous sequences that look the same, and try to get pieces of viral genomes to see what is actually in the sample. And then those are mapped against existing databases, viruses that we know about, the samples themselves within a specific analysis. And we try to figure out, is this thing a virus or not? And that's a little bit difficult still at the moment. Viruses, unlike bacteria, can't be cultured many times. There's a lot of things we just have never seen before we started doing these lab analyses. And there's a lot of things that just exist and don't really do anything to our health. So we don't really know what they are. And you'll kind of see that in some of the figures I'll show you later. But again, basically trying to figure out what in here is a virus. And then I will get into these different ecological measures later, but we study the virome as sort of an ecological population. So we use a lot of statistical methods and measures that are common in the ecology world, like relative abundance and diversity. So I will talk about those later. The gut virome is established in infancy. So the current consensus in the microbiome world is that we're born without any microbiome. But as you kind of can see by this figure here, we start acquiring our microbes immediately. So as soon as you're born, during birth, you start becoming exposed. What is thought to be the way we end up with our established virome is first through this period in which we have bacteria colonizing our bodies. So either through delivery mode, breastfeeding, environmental exposures, and those bacteria, they're thinking that those bring viruses with them. So viruses can be integrated into bacteria, temperate viruses. They can replicate with bacterial DNA, or viruses can replicate within the cells, sort of hijacking the cell machinery and then lyse out of cells. And so what they think is happening is that these bacteria that are starting to establish the bacterial microbiome are bringing phages within them, within their genomes, that are then activated to replicate within the cell and are detectable, but are also detectable just by, everything is homogenized into a sample, and so you end up with bacterial DNA, so you get some of the viral DNA out that way. I'll get into a little bit later what these specific viral taxa are, but viruses, just to note, are classified in similar to the kingdoms of life. So you have your kingdom, phylum, class, or family, genus, species. So there's different phases, or this development characterized by different taxa. In that first phase, there's this bacteriophage of the class Caudoviraceae, which is my Spanish pronunciation of that. That's mostly the dominant taxon that we see there, and there's a lot of unclassified viruses underneath that, but we know it's sort of this bacteriophage class. And then that starts shifting as we become more exposed to different types of bacteria, especially during this about 4 month period. There's a lot more bacterial diversity as we start interacting more with our environment. There's a higher proportion of lytic bacteriophages coming in, both with those bacteria and in response to those bacteria. And we start seeing eukaryotic viruses come in. So those aren't really prevalent in large numbers until that sort of 1 to 4 month range. And there's a shift then from this one type of bacteriophage to this other type of bacteriophage called Microviridae, which is a family, and these other phages that were somewhat recently discovered appear as well. So I'll revisit that in a little bit to show you what our data looks like. A lot of things shape the gut virome. So this is not surprising if you've read any bacterial microbiome literature, but some things that are relevant, especially to the infant virome, are mode of delivery. So there have been a couple studies that have noted vaginal delivery is associated with a higher viral diversity, likely paralleling a higher bacterial microbiome diversity, which is also seen compared to C-section deliveries. Breastfeeding is interestingly associated with lower number of eukaryotic viruses. And the hypothesis for this is both that mom is transferring antibodies, which are directly for eukaryotic viruses, especially pathogenic ones, but also they are, I forgot when I was gonna go there. It'll come back to me in a second. Less environmental exposure. There we go. So the environmental exposure, if you're not breastfeeding, you can get other eukaryotic viruses, which are more prevalent in the environment. And you can see also environmental exposures can mean other things. So babies putting things in their mouth, seeing other people, geography, other aspects of diet as well. So other foods and liquids are a source of plant viruses, which can often be detected in stool samples. So you might not expect a plant virus inside of the intestines, but you find it, usually because of food. Medications also are important to consider in these analyses because we know antibiotics disrupt the bacterial microbiome. And so as the bacterial populations shift, we sort of see this like quick emergence of a lot more viruses as those bacterial cells lyse, and then sort of they die off as well as the bacterial populations decline. So there's definitely shifts that occur with antibiotics. And I haven't seen a ton of good data. I might need to scrounge a little more in the literature, but I think that there's not quite a great understanding of the other antimicrobials yet specifically, but that's something also that we expect would affect Virome. And then one important thing I want to point out as well about viruses in the immune system is there's some hypotheses that viruses might support gut mucosal integrity. And so they have found viruses that sort of attach themselves to the gut mucosal cell surfaces and act as a barrier against bacteria. So it helps prevent bacteria from getting into the bloodstream. Also helping sort of just regulate inflammation and other aspects of that gut mucosa. And so there's some idea that there are viruses that's their purpose, and in some ways are actually might be leaning towards the beneficial side of the spectrum. And last note here is that the virome is highly individual specific and it remains stable over time and actually much more stable than the bacterial microbiome, which is interesting. Not entirely clear why, but it does seem to be more stable, although it will have, again, disruptions from many of these factors. As I was kind of mentioning, there's a lot of ways viruses can interact with our health. So there's the obvious ones that we know they're infectious viruses. Those are eukaryotic viruses because they affect our cells. Again, that idea that some of our viruses might actually help protect us from other infections, other viruses, other bacteria. But importantly, there's a lot of effects of this interaction between viruses and bacteria and inflammation in our immune system. And so this is a nice figure from a great review about the virome and its development. But there's an idea that this interaction between these phages and the bacteriophages might be influential on the levels of inflammation and gut immune development and maintenance. And so this is relevant for HIV in the virome because we know HIV is associated with increased gut inflammation and permeability. And there are not a lot of studies on HIV in the virome right now. It's somewhere under 20 still, and in various compartments and populations. But the sort of consensus seems to be that HIV-associated immunosuppression, low CD4 count, not having been on ART, is increasing the relative abundance of eukaryotic viruses, so viruses that have the potential to infect our cells. Again, not really clear where that's going yet, but that's something that's been consistently shown across a handful of studies. There's also an interesting study from last year that looked at specifically individuals living with HIV who received integrase strand transfer inhibitors, of which one is dolutegravir, which is what Kenya has shifted to lately for the prevention of maternal to child transmission and long-term ART. And in that study, they found compared to ART naive individuals and HIV negative individuals that they had sort of similar gut ecology to the HIV negative individuals, which suggests that ART is helping with immune reconstitution and might be helping avoid some of these issues associated with virome and gut permeability. Now, why do I bring all that up? It gets us to sort of why I'm studying HIV-exposed children. So thinking the current field of HIV, we know there's really great measures to prevent mother-to-child transmission. So we have this growing population of HIV-exposed uninfected children, which I'll refer to sometimes as HEU children. And studies have observed pretty consistently throughout the HIV pandemic that they have higher morbidity and mortality than their HIV-Unexposed and uninfected or HEU peers. There's a lot of reasons this is hypothesized. This is a figure adapted from a really nice review on HEU infants that sort of tries to get at the complexity of these different factors. So there's your socioeconomic environmental factors associated with HIV and health. There's maternal health status, so mom's HIV viral load, her CD4 count, what ART that infant's exposed to, her immune system, her inflammation levels, a lot of which are very relevant during pregnancy and the exposure that the infant is receiving during pregnancy, but then also things that stem off of that and for the infant. So HU infants have been observed to have some immune dysregulation, higher inflammatory levels than HUU infants, suggesting there's something about their HIV exposure, and it's still a little unclear what. It's likely just a combination of these things that is causing that in some infants. And all of these things can interact with each other and lead to increased susceptibility for morbidity and mortality. And what we know then about the virome and about how it affects the gut mucosa, what we're starting to see from people living with HIV is that there's a lot of overlap in things HEU infants experience and what affects the virome. And so that leads to the question of, does HIV exposure affect the infant virome? And that's where we are. So that's my aim, is going to first describe the gut virome in HEU and HEU infants in a cohort that I'll describe momentarily in Kenya. And then also compare the gut virome ecology between those two groups. And what's exciting about this study and why Linda Kizazi is such an interesting cohort is this is the first time we've looked at virome in HEU children at all. Infant data are common, but still pretty sparse. Virome data in general is pretty sparse. A lot of existing data is also from the Europe and the US. And so it's nice to have like this extra longitudinal body of data to contribute to the virome space as well. So the Linda Kizazi study, as Jen said, I've been part of the study for a long time. It's actually still ongoing. It's shaped, it has different shapes now as it's going on. The kids are passing 5 very quickly, which is scary, but you know, time goes fast. But what I'm focusing on is data from the first 2 years, the original sort of follow-up period of this cohort. So we recruited ultimately 211 people from a maternal and child health facility in the Mathare North neighborhood of Nairobi, which is a very densely populated urban area. We recruited them between December 2018 and, as you can see, March 2020 cut off our enrollment because of COVID. And we were aiming for as close to 50-50 women with HIV and women without HIV. So we didn't get quite there, but pretty close. We enrolled adults who were in their 3rd trimester of pregnancy, planned to breastfeed, and if they were, importantly, living with HIV, had received ART for at least 6 months prior to enrollment. And then we excluded women who had any sort of serious chronic medical conditions, had planned a cesarean section, rather than maybe had one ultimately, and then had ever used immunosuppressive or antimicrobial drugs based on these timeframes, with the intention of trying to get healthy women with not like any expected virome disruptions. Now that's a very it brushes over some things that obviously could happen, but especially with these drugs, it's important that we didn't have that at that enrollment time. And then on the right side of this figure, you can see we followed then women from their enrollment in 3rd trimester through 2 years or 24 months. There were study clinic visits about every 3 months. That 1.5 months will be week 6 in later figures, and that 2.5 is week 10. And that's coinciding with vaccination visits, which was helpful to get people coming to the study. And what's important for this analysis is that top bar there, where we are collecting stool samples at each of these visits that are shown in the blue, and that's the samples that ultimately contributed to this analysis. There's other stuff about Linda Kizazi, I'm sure many of us are happy to talk about all these other things, but this is the relevant for our analysis here. Wanted to note when talking about this cohort that we did not see a higher risk of morbidity and mortality among the HU infants in this study. So this is data from my first dissertation aim. And that was looking at the risk of morbidity and mortality over the first 2 years of life in all infants that had follow-up data, which was a population of 187. And these are the survival curves showing diarrhea, pneumonia, and any respiratory tract infection is what RTI stands for. And what was really interesting is that we saw the opposite in respiratory infection of what we'd expect. So the consensus is HU infants have a higher risk of everything, and we actually showed that they're not having, they had a lower risk of pneumonia and any respiratory tract infection. And we hypothesized that this was because a substantially larger percent of the HU infants were exclusively breastfed for 6 months than the unexposed kids. And we know exclusive breastfeeding is associated with positive respiratory health outcomes and also diarrhea, which might explain some of the offset. It's a very interesting curve. I'm not going to spend too much time on it. But just to note that we're not seeing sort of that general trend that has been experienced in the past. We can talk more about maybe why later. For stool samples, so the stool is collected either by a study nurse or the infant's mother in a sterile collection container. We had a really great lab and clinic team that got everything to the lab really quickly because you don't want stool samples sitting around, because what's interesting about stool samples is all the viruses and bacteria will keep doing their thing in the sample. And so it will change if you let it sit out too long. So we had a great pipeline of getting that to the lab so it could get stored in the freezer as quickly as possible. And those were stored at -80 degrees until the sequencing, which was done by a partner laboratory at Arizona State University. For this analysis, virome testing and microbiome testing are very expensive and labor intensive. And so we selected a subset sampled a couple years ago. So we had samples prioritized between January 2019 and December 2020. We tried to prioritize infants with vaginal delivery because we know cesarean sections, which have a couple like emergencies happened. So we tried to restrict just the vaginal deliveries to avoid that confounder. And then we prioritized infants that had more complete sampling or samples in year 2 so that we could see longitudinal data. And as I sort of mentioned before, we studied the DNA viruses. So all of the data here are just DNA viruses. So a limitation of this is that we don't have any RNA virus data, but that is pretty consistent. From an analytic approach, there's, as I kind of mentioned before, we will look at ecology measures and compare those between the HU and HU infants, and I will explain what they are right before I show you the data, so it's not as, they get confusing sometimes. But the description is sort of understanding like what's there and how much of the viruses are there, and then the comparison is comparing those metrics and then also looking at this thing called beta diversity, which I will explain. And you may ask also, why is an epidemiologist doing a study? And it's a really interesting space. A lot of lab studies don't necessarily have the same lead up into a study design and thinking through all of these different possibilities for confounders. And it's because lab work is super intensive and I have learned to have a lot of respect for lab because I have no idea what you're doing at the time. But it's been a really great learning experience because I think there's a lot of mutual benefit to working in a space where you have your epi training, and then you can help apply it to some of these other studies to strengthen them, and then lab folks can tell you why what you want to do is impossible. Expensive. But it's been a great learning experience, and really has been wonderful for my own thinking about biases and everything. Okay, so study data. Ultimately, we have 203 samples. There were a couple more that got dropped for QC reasons, but our ultimate analysis has 203 samples from 69 infants that were collected between day 4 of life, and there was 1 month 21, but most are through month 18. This table here is showing the infant characteristics. So we had about half and half. There was 54% were HIV-Unexposed, 46% were HIV-Exposed. There weren't a lot of differences, but I do want to highlight specifically the differences in feeding. So as I mentioned on the slide with the data from my other analysis, there was more exclusive breastfeeding for 6 months, which is the recommendation by the WHO and the Kenya Ministry of Health. That was a much higher percentage also in this sub-analysis, this subgroup of participants. So 91% of the HU kids were exclusively breastfed for 6 months versus only about half of the HIV-Unexposed kids. This is sort of consistent with what we kind of heard from other ongoing GWACh cohorts, some other studies from Kenya. So it's not shocking that we're seeing that. a lot of women living with HIV have good engagement with healthcare. There's a lot of messaging of exclusively breastfeed your infant for 6 months. So that's one of the reasons we're hypothesizing that that difference exists. What is also interesting though, and again, saw in the full cohort, is that the duration of any breastfeeding, so not just exclusive breastfeeding, but any breastfeeding is actually shorter in the HU kids. So moms living with HIV are exclusively breastfeeding for 6 months, but weaning around 1 year, whereas the moms who are not living with HIV sort of wean around maybe 1.5 years. So there's a longer duration, and I think that's also consistent with some of the other cohorts that are ongoing as well. Haven't come up entirely with an explanation of why, but this is something that we've been seeing. So the takeaway is that feeding will be important to consider in this analysis. For moms, so this is moms and mom and sociodemographic information. There are not a lot of differences here either. And again, all of this is by design. We enrolled everybody from the same clinic, from the same small catchment area. There's very similar sociodemographic characteristics throughout the neighborhood. And so that was intentional to try to minimize biases from these potential confounders. Here, I'll just highlight that moms living with HIV were a little bit older, also consistent with other studies, and moms living with HIV had more secondary education. So potentially thinking about things that might go into later analyses to adjust for confounding. But as a whole, similar exposures between the exposed and unexposed kids, and this subset, it looks very similar to our cohort of every infant that had a follow-up visit. So that's also encouraging for representativeness. For the moms living with HIV, so one of the strengths of Linda Kizazi is that we have sort of a homogeneous group of kids exposed to HIV. So they all were born around the same time. Were exposed to this, almost all were exposed to the same ART regimen in utero. There's a lot of implementation in this area of other PMTCT methods to help reduce transmission. So infants were getting their antiretrovirals, they were getting metronidazole prophylaxis, and moms were also probably their HIV viral levels were probably suppressed. We unfortunately don't have HIV viral load data, but we do have normal CD4 counts for moms at enrollment. A couple of things to highlight here. Almost all moms were diagnosed with HIV before pregnancy. There were only 2 that weren't. Of those 30 that were, everyone had started ART before pregnancy, with a median of 5 years on ART. So this is also longer term ART, which is a little bit different than some of the published literature. Some of the older cohorts have a lot more heterogeneity in ART experience. This cohort was established right before Kenya switched everyone from efavirenz-based ART to dolutegravir. So we had that switch happen during the follow-up period. But at enrollment, almost everybody was on the efavirenz-based first-line regimen. The others are a little bit of a mix of first and second line, and there's 1 mom on the dolutegravir regimen. So takeaway from this slide is a decent homogeneity also within the maternal HIV characteristics for the HIV-Unexposed kids, but also limits our ability to look at differences within that subgroup because we don't have a lot of differences, again, by study design. Okay, so some results. So we'll start with relative abundance. If you read a microbiome paper, this is going to be those fun, colorful plots with all the bars. Relative abundance is a measure of how much of a sample consists of a specific virus. In this case, because I'm going to be sticking to the family level of viruses, and this is because once you start getting to the genus species level of viruses, the data quality is a little bit iffy because the reference databases are incomplete and it starts getting a little bit less certain. So we typically are staying at this family level. So when I'm saying viruses here, I'm usually referring to members of certain families. But I do know, like from the data, that there are unique viruses to just cluster within families in these figures. But the point of relative abundance is just to understand how common or rare a taxon is in a sample. And this depends on how much stuff is in a sample. So in this little example here, sample A has 9 viruses, sample B has 4, and each of them we see one of the blue viruses. But in sample A, which has a lot more viruses, the one blue virus makes up a smaller proportion. It's just 11%, whereas in sample B, it's 25% because there's less stuff there. So there will be, like, relative abundance is a measure that sort of varies depending on what's already in the sample. One analytic note is that if for those of you who know more about microbiome sequencing, sequencing is not always even, and how deep you sequence is not always even across samples. That has been accounted for in these analyses, and so what you'll see is the normalized data. So this is a very fun rainbow plot. On the left here is the HIV-Unexposed samples, and on the right is the HIV-Exposed samples. And there's nothing super shocking different about these, which is a first pass at going, is HIV exposure doing something to affect the gut virome? So as a whole, not accounting for feeding, not accounting for time and the development of the virome, we're not really seeing anything super different. But to walk through a couple things that we're seeing, which are interesting. So this big red wedge at the top of these is this abundance of these unclassified viruses that are all bacteriophages. And this is something we expect to see. So if you remember back to the beginning when we were talking about virome development, those are those original sort of like the foundational viruses, the cohort. What we can't really see from this figure because it's organized by the actual relative abundances is what's happening over time. But what we do see again is we see that high abundance of those viruses, which is we expect. Anything in red here too, so in the red boxes, so you'll see sort of that really dark red, that lighter red, and then that yellow through blue are all different types of bacteriophages. So the takeaway from that is that the infant gut virome is very predominantly bacteriophages, as we expect. These two, the orangish ones, are both eukaryotic viruses, and those Anelloviridae are this eukaryotic virus that we acquire sometime in that 1 to 4 month range of our virome development, and they are ubiquitous. Like everybody has these, and we don't really know what they do. They just sort of are along for the ride. And so there's a lot of work right now to try to understand, like, what are those viruses? Are they conferring a benefit? It seems that the literature has ruled out a pathogenic effect, but that is interesting that we're seeing those here. And then again, this is expected. This plot is getting now more at the idea of time. So these bars are representing study visit time points. So day 4 is on the left, month 21 is on the right. Take month 18 and month 21 with a grain of salt because the sample size is very low. It's only 1 on month 21. But what's interesting about this plot, so again, thinking about that shift from this Cohort class to this specific it's actually under a different class, this Microviridae family, that sort of lighter red, you can kind of see that happening. So you can see that big red, dark red wedge of those Cohort declining over time. And that other red color is starting to like increase a little bit. Again, it's hard to see because there's so much going on, but you can sort of see at a glance that is shifting. And that's exciting to see as well, because again, it's consistent with the other literature on virome development. Our data are looking good. And it gives us sort of a window to see what else is going on there. So like I said, those are all actually within the same class. So even those lighter bars are part of that same class of bacteriophages. And then I'm pointing out these 3, that yellow through green. There are these viruses called crassphages. And I mentioned at the beginning, they're sort of a recent-ish discovery in the virome world. And there's still a lot of like, what are these things and what do they do? They're bacteriophages, but what's pointing them out because these also start increasing in abundance over time. And so it's a little bit hard to tell because there's, again, so much going on. But there's a little bit more yellow as you get a little bit farther down. So that's also sort of reflecting these shifts that we expect. So once we look at relative abundance, gives us kind of a picture of what's there, how much is there, but it's hard data to work with besides those figures. So we go next into this metric called richness. And so richness is a count of how many viruses are in a sample. It's just like purely how many. So in our examples from sample A and sample B again, both have the same 3 types of viruses. So we would say both samples have a richness of 3. So the limitation of richness is that it doesn't account for how much of something is there, but it can still be interesting to know what is there because that might track with different changes over time. So this is a figure stratified by both HIV exposure and time. And so HIV exposed kids are that blue color, HIV unexposed kids are the red color. And what we're seeing is sort of this between day 4 and week 10, which is not about month 3, is sort of this lowish level of number of viruses. So that the y-axis here is going to be, you can think of it as like a number of viruses. So you see sort of this lowish number, they're similar, and then you see this big jump around month 6. And this is, we're hypothesizing that this is related to the introduction of new foods. So around month 6 is when kids, especially those who are exclusively breastfeeding per recommendations, are starting to have other foods and liquids. And so that starts introducing all sorts of new viruses and bacteria. And so it's pretty expected then that you would see this big jump in the number of different things that are in the virome. And that is indeed statistically significant. When you compare each of these time points, when you compare HIV exposed and unexposed within the time points, none of them are actually statistically different, but this is using very basic statistical tests. So take that with a grain of salt because there's a lot of confounding and other things that are not, that's not incorporated in that, but that is the biggest significant shift that we're seeing in richness. Similar to richness, but a little bit more complex, is the measure of alpha diversity. So alpha diversity, this is one of the ecological metrics, intends to be a little bit more descriptive than just richness alone and add in that relative abundance component. So it's not only telling us what's there, but how much of something is there. And it creates this index. So there's formulas, there's a couple different ways of measuring alpha diversity, but you basically plug these things into a formula and you get some sort of index number, which is a continuous number that describes the population of viruses within a sample. And so we say it's describing within sample diversity. When we talk about beta diversity, it's going to be more about a comparison between 2 samples, but you can think of alpha diversity as just, this is describing one sample, it's describing the number of things and or the population of that sample, and it's giving sort of like a number to that characteristic. So we can plot that similarly to richness. So the index that I used is the Shannon index. This is probably the most common one that you see in the literature. And it's this continuous number from 1 to 6 on the y-axis. And you can see, unlike richness, there's not actually a ton of variation over time. There might be there's like some separation happening within time points here and there, but overall it's kind of all in this like middle 3-ish range. And when you do those those within time point tests for HU and HUU, there's no statistically significant difference, although there's some trending happening at day 4 and month 9. But again, taking that with a grain of salt because not considering other factors that might be influencing things is not counting for sample size either. And there is some variability in HU versus HU sample size over time. But this also isn't, when you think back to the relative abundance plots over time, it's not entirely too surprising though. So if you think about, you have like all these bacteriophages, right? There's so many different bacteriophages and there's just high abundance of all of them. And so if you throw a couple new viruses in there, some of the eukaryotic viruses perhaps, and you're seeing those appear with the richness, right? We see that jump at month 6. But there's still this huge dominance of the bacteriophages and these common ones that we expect to see. So I'm not as surprised that alpha diversity doesn't shift because there's just so much of the same thing existing from time point to time point. So that's one of the reasons I'm thinking we're not seeing that same jump up in diversity that we're seeing in richness. So there's new stuff, but it's not dominating the virome. And then last, we look at beta diversity. So beta diversity, and please interject if you have questions, because it took me a while to wrap my head around this one. But beta diversity is comparing samples. So you can think of it as a series of pairwise comparisons between one sample to all the other samples in your sample set. And that repeats for every sample. So every sample has these relationships with other ones. And there are a couple different ways of measuring that difference between those samples. Usually we're looking at difference rather than similarity. There's a lot of fancy math that goes into these metrics that I will spare everybody, but basically there's a way to enumerate how similar or different something is. And that's what we end up getting with beta diversity. And what I think is helpful to see is just what that data looks like. So unlike the continuous outcomes for richness and alpha diversity, we're not seeing like one sample and then one measure, which is sort of what I think especially epidemiologists are used to seeing. Instead, we have this matrix. So we have these pairwise comparisons, and each number here is one of the beta diversity measures. So these are distances, Bragg-Curtis distances for those of you who like ecology. And so you'll see the bigger the number is closer to 1, it's going to be more dissimilar. And if it's closer to 0 or is 0, as you'd expect from comparing the sample to itself, it's going to be that low distance. And so you can use this matrix to do analyses. But again, not so much just like a single number per sample, but you get this giant table of comparisons. And so the way we look at those is using this analysis called principal coordinates analysis. And so basically what that analysis does is takes all those different comparisons and tries to figure out which ones are the most important, which ones are the most influential for where these points or where these samples are relative to each other. This is where the conceptual part is a little bit tough. But what these plots boil down to is you have on your x-axis a number that's describing like the most important distances in that matrix, the ones that seem to describe how the samples are similar or different to themselves the most. And then that second axis is the same idea, but sort of like the second tier of importance. And we plot them against each other to try to see where samples are clustering together, because clustering means the samples are probably somehow similar to each other. And we want to see what makes them similar to each other so that we can think a bit more about the biology behind it. So in this plot, again, the HEU kids are the reddish color and the exposed kids are the bluish color. And this plot on the left is very unexciting. Everything is overlapping, which is suggesting that there's not really any clustering happening by HIV exposure. HIV exposure is not really seeming to be why things are different from each other. The same thing is on the right. So the difference between the left and the right is that on the left, we're just comparing whether viruses are there or not, so presence or absence, thinking of it like richness, where it's just is what's there. And on the right, we're accounting for how much of each thing is there, and it uses relative abundance. And so we see that there's some weird clustering happening, but it's not by HIV exposure. So when we look by time, we see a little bit different. So instead of each study visit, because the sample size isn't enough, I've clustered them together, or I've grouped visits together. So the red is visits before 6 months, green is visits between 6 and 12 months, and then blue is visits past 12 months. And those divisions are kind of based on sort of that jump that we were seeing in the richness, and also sort of what we expect when the virome is shifting in composition. And so on this left plot, we see that this under 6 months group is clustering together a little bit more away from these other visits. And what that's suggesting then is that there's something about what viruses are present in that earlier time period that makes them different from these viruses that are present at the later time period. And I guess I should say virus is like virome as the whole, right? So it's not just like specific viruses, but the whole community within the sample. So this is just, again, considering what's there and what's not. This plot, when we start taking into account how much, that separation sort of goes away a little bit, changes actually, and gives you sort of this ellipse happening over here with the later time points. This is a little bit, we need some more digestion of what's going on here, but we can see probably from these tails too, like this is definitely the ones over here are getting clustered into this 12-month time point, and these are all green. They're all outliers, but they're all green. So there might be there's some time clustering happening is the takeaway from this. And that's not surprising. We know the virome changes over time. So we would want to see something happening based on time. And that's where the PCOA plots go, sort of like a descriptive way of maybe seeing something. Okay, so key takeaways. To summarize all that, so we're seeing the viruses we expect to see at the times we expect to see them. we're seeing that really interesting jump in richness around 6 months, which we think might be due to that new foods and liquids. But we're not seeing that in the alpha diversity. There seems to be similarity over time. And also same with the beta diversity when looking at HU versus HUU. So if you kind of had to summarize the data at the moment, I'm not seeing a strong signal that HIV exposure is driving any differences in virome between HIV-exposed and unexposed infants. Something else probably is, aside from, and there's age, there's feeding. Sorry, I didn't have the feeding data today, it's in progress. But there are other things to look at. We want to see if there's anything in our data set that might help explain how virome changes. Some brief strengths and limitations. So as I mentioned a couple times, the cohort design really was intended to have a homogeneous group of HIV-exposed infants and a comparable group of HIV-unexposed infants, and we did a pretty good job with enrolling that. Relative to existing studies, this one's pretty big. Virome studies are small for the aforementioned time, money, intensity of doing the sequencing, and so this is a good chunk of data to contribute to the field. We also have good visit level data for key factors that influence the virome, like feeding, antibiotic use, illnesses. Again, sorry, none of the data is in here. Stay tuned. And we had a lot of methodology that was consistent, again, with published literature. So we expect our results to be comparable to what's out there for that reason. We did not, of course, include all the infants in the cohort and all samples from all the infants we had. A lot of that's logistics, so that there is still the possibility that there could be differences if we looked at everybody, but we did have a seemingly representative subset of everyone based on the background characteristics. Only DNA viruses again, but this is also consistent with existing literature. It's pretty tough to get the RNA viruses. And we didn't have that heterogeneity among the HU infants to look at differences within HIV exposure, which by study design, but also is something that people usually want to know. So unfortunately, we can't really look at that here. And this is a healthy cohort. So with good exclusive breastfeeding among the HU infants. So there is the consideration that this might not be generalizable to other populations of HIV exposed children. So just noting that for the bigger picture. And next steps, so the next step is to do some more rigorous modeling for those diversity outcomes and richness. So I'm going to use some GE models adjusted for potential confounders. This is the epi part that gets to come in. There's a method called Hermanova that is used for beta diversity. So it can take that matrix and essentially do modeling on that and account for some of these other factors as well. So we'll get some data for beta diversity too that are just more complex than the sort of pairwise testing that we've been doing. And then we'll also look and see, like, are there any specific viral taxa that are more dominant in one group than the other? There's a lot of different stats methods for doing this as well that are popular in the bioinformatics space. And that's sort of a typical part of one of these analyses. So we'll see if there's anything specific shaking out there. And then repeating, again, all of this with other groups that might help explain some of the virome development, or that would be good to consider. So with that, I want to really extend thanks to all of the study participants, our staff, the amount of complexity that this study has had in logistics, and especially the sampling. We have tens of thousands of samples in the study, and so a huge thanks, especially to the lab team, and just could not do this work without all of the time, dedication, and effort from our staff and our participants who really have been with us for a long time, and we're really grateful for all of that. And a big thanks to all of our other collaborators as well, and all the mentorship they provided me for quite a few years. So with that, I think I've left us about 10 minutes for questions. I have my email down there too if anyone online is listening and wants to send anything that way. So yeah. Drink some water.
I have many questions, but I want to hear questions from the room and the online audience maybe first. Scott, I know you have a question. Your picture is at the top of our screen, so I'm going to pick on it a little bit. I know you work on microbiome and have done those studies and are familiar with this area, so
I know.
I hope you're excited.
I am excited. I'm struggling still to know exactly what the DNA virome means. But the questions I had were actually more specifically related to the Bray-Curtis principal components plots, and I'm wondering if you could go back to those.
Do you prefer the visit ones or the HIV exposure ones?
I had, I think, similar questions about both. This one is fine. So this was by HIV exposure.
Yes.
And the first question I had, because I've come across this and I don't really know what to do with it myself, is when you had these sort of best fit ovals plotted onto it, often there's a way to do that with a normal or a t-distribution. Do you know which one you used?
That's a great question. It's the default of whatever is in R, so I will have to look. But I'm guessing it's definitely not something that I
I
altered into
think
matching
the
my
default,
data, so it's
I
close.
think the default is T.
Sure, and
Um,
that makes sense.
this is something to think about though, because I've had, in this case, if your t distribution isn't showing anything and it already is weighting the outliers, then you got nothing. But I've had situations where the t distribution looks like there's something going on and the normal distribution doesn't. And I still don't know exactly what to do with that. The other big question I had with these is that when I've seen microbiota data in a principal component plot, shape the way that it does in your weighted Bray-Curtis plot on the right, so that it's a little bit triangular. Typically, if you look at the specific viruses that are identified in each of your individual points, it's possible to sort of create like vectors for what each of those, what groups of bacteria are highly represented at each of those points. And to give a very specific example from the vaginal microbiome so that you have an idea what I'm thinking about, it creates a triangle typically, and you end up with a vector that's the Gardnerella morphotypes, a vector that's the L. crispatus morphotypes, and a vector that's the L. eners morphotypes. And that's where most of the observations fit. And I've found it really helpful in looking at these principal component plots to label what those vectors are so that you understand what bacteria, in this case viruses, are populating out at each point so that they're not just sort of abstract points. And I'm wondering if you know at the points, and it'll help you to know that I'm using my pointer to point, which of course you can't see at all. But at each of the points, so that the upper, the right hand, and the lower point of that sort of triangle, what's represented there?
Yeah. Yeah, I really appreciate that because those tales have been frustrating and I haven't had a lot of opportunities to dive into it. I think I asked a couple of questions to our Virome experts and they were, well, something else might cluster it and you need to keep figuring out what it is, but I like the idea of looking more specifically in this way at like what's actually in the sample. And it's interesting to know that you've seen that in the bacterial microbiome as well, because it really is this triangular sort of thing here.
Yeah, I have a very recent manuscript that's actually under review that I can send you that just shows exactly what I'm talking about. And you can actually go in point by point and disaggregate what each of them is. And then you understand, like, what does it mean to have a high score in principal component 2 and a low score in principal component 1? Like, what are the, because what interests me partly about this is that there are a couple of small but interesting groups here, but neither of your circles encompasses them.
Yes.
And to me, it's the group at the far right and the group at the bottom, but compared to the group at the upper, I'm sorry, yeah, the upper left to the lower left and to the far right.
These guys, they're really, yeah, they do seem to have 3 clumps there.
Could you go to the one that does the by time? Yeah. Yeah, it definitely looks like the
Time is
time is a better discriminant of diversity than.
Yeah. They
Just
still
explaining
outline,
words.
because these ellipses are like
Yeah.
the 95% confidence interval that you
Yeah.
can think of as
Yeah.
is what's grouping there, and so those are still outlying, but you're right, they at least do
They do
cluster
a
at
better
a
job.
time.
Yeah.
Which I wonder when I look at this by feeding too, what I'll see, because feeding and time are so correlated in this cohort. But I'm not convinced that's gonna be like perfect either, so. But yeah, I'd love to see that paper if you don't mind sending it. Thank you for.
Can you very quickly remind me of your email address?
It's ERB29.
Got it.
I have a question about these plots as
Yes.
well. So you're using Virome as an outcome in this analysis to query like what are the main determinants, right? And it looks like HIV exposure is not coming out as
Yeah.
being like particularly like
Yeah. It
big
doesn't seem to
for any of the ecology indices that you've looked at. Can you use these same methods, like the plots and the Bray-Curtis distances, to look at Virome as an exposure? To look at then look and see like say like an outcome of like intestinal integrity or environmental neuropathy is like the clustering, I'm trying to wrap my head around like also the nuances of like how beta diversity, right, the interdependency and it being a matrix, like how you think about it differently as an exposure and outcome.
It is, I will, and what my understanding at this point of beta diversity is not easily boxed into my usual epi framework of exposure outcome. It sort of is like its own thing of, so this matrix that you have, like if you wanted Virome to be your exposure, like you still would have to kind of look at it backwards. Like you would still look at it in the same way where you want to see the clustering, but by those those other indicators. Now, if those other indicators are continuous, I have no idea what to do. That's outside my current scope of it. But if you had like yes or no, environmental enteropathy, for example, then you would still end up with the same sort of thing. So that's where it's not quite the same like exposure outcome measure, and that was something I did not realize before. Whereas in the richness diversity thing, you could kind of flip those more easily because that's more typical data. Whether you want to or not is more of the methods question. But you could look at it.
Okay, about 1 more minute for one more question before we have another group coming in.
Can you hear me? Hello, Emily. That's a good.
Hi.
I have a quick question for you. Have you looked at the phages that you have earlier on? We know that phages go through different cycles. Some are lytic, some are lysogenic. Have you looked to see whether between your groups there's difference in those phages that are lytic versus those that are lysogenic in their life cycle between HU and HEU? That is one, to see whether there's a difference in what life cycle the phages prefer and whether HIV exposure impacts that. And number two, have you looked at gut microbiota, the bacteria side, to look at the relationship between the bacterium and the virome, just kind of to look at how are those changing over time and thinking about predicting, since you have phages, what are some of the predicted hosts or some of the bacteria that those phages can actually infect? Have you thought about all those in your data sets? Otherwise, great talk.
Thank you so much. I like the idea of the lytic versus lysogenic phages. I wrote that down to think about a little bit more. I've not done that, but I like that idea. For the bacterial microbiome, we have some bacterial microbiome data within the cohort, but it's not overlapping perfectly with the sample set. So that's currently outside the scope of this analysis. But ideally, we will at some point find a way to look at the bacterial microbiome. And I assume at that point combine some of this data, because that is exactly something I've also thought about wanting to know is what's going on with the bacteria, because there really are a lot of they're interacting. They're not independent things, and so knowing what one's doing and what the other one's doing would be really, I think, helpful for understanding sort of the more of the biology part of why we see the virome doing what it's doing. But Unfortunately for the dissertation work, it will not happen, but maybe in the future.
It's a great postdoc.
Yes, always looking for more ways to prolong this. Yeah. But thank you, I appreciate the question.